Great. Okay, so it's a real pleasure to be here and to be able to present the work that our multi-investigator project did, funded by the BRCCH. And this was many multi-investigators, so some of them are here today, and maybe some of you would like to wave if you're, so people know they can also answer questions. So Mederick Diard is here, Sean Froese is here, Viola Vogel was going to be here, will be here later. So there are also other PIs who I will mention throughout the talk. But what we set out to do was really to take some fundamental progress that we had made on engineering or coming up with ways based on oral vaccines and microbial ecology to engineer microbiomes and to put this into some clinically relevant situations. So the first thing, of course, is it's great to be able to engineer cool things and to be able to build cool things. But if you build cool things in a way that's not useful, it's not great for anybody. So we need to actually know what we're trying to do here and what we're actually trying to achieve with the strategies that we're developing. And I want to tell you about two stories that we worked on. It's actually part of what we did in the project. The first one is about optimizing microbiome function to ameliorate urea cycle disorders, and I'll explain what those are in a minute. The second one is about eradicating neonatal colonization by pathogenic E. coli. So they sound very different strategies. They turned out to be very different strategies. We started out thinking we would be able to use the same thing on both of them, and you'll see as I go through why that was not the case. So there's been a lot of work done on how your microbiome helps you to assimilate carbon. So carbon coming from sugars, and you break down this carbon, you get short-chain fatty acids made by these bacteria. This gives you more energy from your food, and that's a great thing. It's been heavily studied. There's been much, much less work done on nitrogen. So obviously we also need nitrogen. You need nitrogen in all your proteins, nitrogen in all of your nucleic acids. But you also need to be able to dispose of excess nitrogen from your body. And particularly when you break down all of those compounds, the waste product you produce is ammonia. And ammonia is extremely toxic in too high quantities, and it's particularly toxic in brain. So the reason for that is you put actually quite a lot of effort into detoxifying ammonia in the liver. You have the urea cycle, so this goes between the mitochondria and the cytosol of hepatocytes. You fix ammonia through this cycle into urea, and that urea is excreted via the kidneys and actually also via the intestine, underappreciated role of the gut in urea excretion in humans. But you also have to detoxify ammonia directly in brain. It's a very energy-consuming organ. You produce quite a lot of ammonia in brain, and there it's actually detoxified by fixing it into glutamate, producing glutamine, which is then exchanged back into the circulation and disposed of via the liver. On the other hand, you have bacteria in your intestines that can do much more imaginative things with ammonia, right? To bacteria, ammonia is not necessarily just toxic, it's also something they can use as a nitrogen source. Most bacteria are capable of assimilating ammonia into amino acids and nucleic acids, but they're also capable of doing the reaction in the reverse direction because microbes are a bit more flexible than we are metabolically, generally speaking. So this is already looking like quite a complicated system, and what happens very rarely is that humans are born with a mutation in the urea cycle. And the most common urea cycle disorder is actually a mutation in this gene called ornithine transcarbamylase. The reason this is the most common is that it's an X-linked gene, so boys born with this only require one copy of the mutation. And if you're unlucky to be born with one of these mutations, you have a major problem detoxifying ammonia. And actually, if you have complete loss of ornithine transcarbamylase function, this is typically perinatal lethal. So as soon as you don't have access to your mother's liver, your own body cannot detoxify ammonia, and you very, very quickly go into hyperammonemic crisis, coma, and this is very, very difficult to treat. But there are quite a lot of cases where you have hypermorphic mutations. So you have mutations in the gene, it's still there, it still functions a little bit. And those individuals are diagnosed typically a little bit later, so either within weeks of birth or actually up to individuals in their 40s are sometimes diagnosed with these conditions. And what these individuals, particularly later in life, present with is typically developmental delay, so they have long-term chronic brain damage from this continuous exposure to mild hyperammonemia. And they will develop hyperammonemic shock and go into coma if they go through any sort of metabolically stressful event, or often during an immune response, because immune responses also generate a lot of protein breakdown and a lot of ammonia. So the clinical consensus in this disease and in doctors treating this disease is that the microbiome is a metabolic burden in urea cycles, because there are bacteria present that can break down urea and they can release ammonia, and that contributes to the ammonia that the patients need to detoxify. And that's somehow difficult to treat, difficult to deal with, and therefore actually a challenge medically. And so we were brought in to say, can you engineer the microbiome of these patients so that we don't have this problem with it being a metabolic burden? And initially we said we have no idea how to do that. But we then went back to them and said, okay, well, if we don't look, we'll never know how to do it. So let's do a project and actually try and work out how this should be done properly. And the first thing we did was we took their animal model of a urea cycle disorder. It's a relatively mild model called the SPF-ASH mouse. It contains a mutation in a region of the OTC gene that affects splicing, so there's some residual function. And we took those mice and we derived them into germ-free conditions. So that means they have no microbiome at all. And when we did that, what we found is that, contrary to what we were expecting, the clinical concept was that if we did that, there'll be no microbiome. These mice would be perfectly, wonderfully happy because they don't have this burden of ammonia coming from their gut. And actually what happened is that if you take these germ-free animals and you feed them very slightly more protein than usual, they actually very rapidly go into hyperammonemic crisis. So they have much, much more trouble handling protein than a urea cycle disorder animal that has a normal microbiome. So this is just weight change over time of those animals given a very small protein challenge. So when we looked at this data, we were initially rather surprised, and we went back and checked that they really were germ-free and all the other things that you would do when you get something that is surprising. And then realized, okay, to understand this, we're really going to be able to quantify what is happening with the nitrogen handling in a condition where you do have a functional microbiome compared to the condition where you don't have a functional microbiome. And that sounds all very straightforward, and we launched ourselves into this, but actually it turns out that measuring ammonia is particularly challenging. It's one of these very small, very common compounds that it absorbs to everything. It's extremely hydrophilic. It has, ammonia versus ammonium. There's a lot of colorimetric kits that detect all primary amines, which are not very useful. And so we ended up developing an LC-MS technique as well as some software to allow you to actually do targeted analysis of simultaneously 50 amine-containing compounds, including ammonia, in all sorts of body fluids. And this is now working very nicely and let us do quite deep analysis of the phenotype of these animals. And what we observed very quickly is that if you look in the intestinal content of germ-free animals, actually pretty independent of the genetic background of the animals, they have extremely high levels of amino acids. So germ-free mice have less ammonia. That figured, that worked with what the clinicians were expecting. But the levels of these amino acids are around 10-fold higher than you would see in colonized animals. And it's pretty much all amino acids. And actually, on top of that, that's a concentration. And when you don't have a microbiome, actually, you have a lot more intestinal content because there's a difference in fluid handling in the intestine. It's actually very similar when you take antibiotics. So in fact, this equates to a roughly 100-fold higher exposure to amino acids in large intestine in animals that are germ-free. And if you then look at what's going on in the brain, because we know that's where the first pathology comes up in these animals, what we find is that you see the same amino acids highly overrepresented in brain of animals carrying this genetic deficiency. So they seem to have a deficiency really actually not only in from the microbiome in breaking down or using these amino acids in the gut, but also in getting rid of them from the circulation. So you have accumulation of amino acids in the circulation, also then high levels of ammonia in brain, high levels of glutamate and glutamine in brain, and that seems to be driving the pathology that is associated with having no microbiome in the germ-free conditions. And you can see if you compare animals here with the genetic deficiency, but with a microbiome or without a microbiome, we have more severity in the animals that don't have a microbiome. So then, of course, the question was, so what can we do about this? Is that useful information? Can we actually use that to treat the patients? And And the assumption is that actually really what's going on here is that when you have bacteria present in the intestine, they're using amino acids to grow. So they're simply fixing those amino acids into biomass. They're no longer available to you as a host. And to some extent, any microbe seems to be able to do this. So if we compare the amino acid concentrations in animals that have no microbiota to, these are two different notabiotic microbiotas with either 12 species or 8 species, those are already pretty much sufficient to recapitulate the phenotype with a normal 500 species microbiome. So that's great news. You can pretty much have any microbes there. We have one microbiome with only 3 species that, for some bizarre reason, vastly overproduces a tertiary amine called cadaverine, which might be interesting. We're going to follow that up. But effectively, what the conclusion of this is for microbiome engineering is that actually having this high fixation of amino acids and ammonia by microbes in the gut helps you to buffer protein exposure. That's probably true whether you do or don't have a urea cycle disorder. It backs up the controversy in clinical management of these patients about the use of antibiotics. So there have been cases where people have said it's helpful to give broad-spectrum antibiotics, and a lot of cases where people have said it's not helpful to give broad-spectrum antibiotics. This seems to back up quite nicely the concept that it's not a good idea to give these individuals broad-spectrum antibiotics. And also the concept that giving them lactulose, which is a microbiome fermentable laxative, so it's going to increase the turnover of bacteria, you're going to be producing more bacterial biomass, is actually quite a good way of sinking amino acids and nitrogen out of the system in these patients. So having started this off about personalized medicine, what we can say here is that actually we can use very broad interventions at the level of the microbiome to help these patients. We don't necessarily need to go highly personalized in these cases, which is great because they're very rare cases and you would want every clinic to be set up, every neonatal clinic to be set up to be able to do this. Okay, but we set out really wanting to be able to do highly targeted, personalized microbiome engineering. And for that, we turned to a different set of pediatric diseases, which actually the infectious diseases driven by Enterobacteriaceae. So these are obvious, nasty things that one doesn't have to have in the microbiome, or doesn't have to have pathogenic versions of them in the microbiome. Bedjo already mentioned this. And actually, if you look at the WHO list of pathogens that are becoming critically or very highly antibiotic resistant, all of these bugs come up in these lists. These are all organisms, they're resident in the microbiome of healthy adults. They can spread either within an individual. So most invasive E. coli infections actually in adults definitely spread from your own intestine to other sites in the body. But also between individuals, particularly during the early neonatal phase, there's transmission from mothers to babies. So one of the infections we were interested in is neonatal meningitis. Neonatal meningitis is predominantly caused by something I'm going to refer to as a K1 E. coli. I'm going to explain what that is a little bit later. But the situation with neonatal meningitis is that in places like Switzerland, it occurs in around 1 in 4,000 live births. In low resource settings, it's occurring in more than 1 in 500 live births, so it's not that rare. And the consequences of this are severe. So in Latin America, Caribbean, Sub-Saharan Africa, and South Asia, more than 50% of cases will be lethal. So there's a very, very high mortality rate. And for those that survive the infection, a decent fraction of those will have lifelong impairment as a result of that. I also just wanted to point out that the same organism causes other infections, so it will also cause neonatal sepsis. It's related to the bacteria that cause enteric infections, and in adults it will also cause urinary tract infections, pyelonephritis, and sepsis. So this is a nasty target that we would like to carry around with us a little bit less. I also just wanted to quickly note here, particularly because I thought Viola was going to be here, but Viola I think will only come later, is that we also did other things in this study that I'm not going to talk about today related to different diagnostics for this and looking at severity and looking at gut pathology that I'm not going to talk about today, but they're all published, so if anybody's interested, do go and read about them. Okay, so now coming back to this K1 E. coli, and E. coli is the bacterium we know most about. Right? It is the best studied bacterial organism on this planet. But the challenge is that we mainly study this guy. So We have E. coli in the lab that we can work on very nicely. And real pathogens out in the world tend to be slightly different. And one of the big differences relates to the presence of what we call capsular polysaccharides. So this is a nice quote from 1947 saying, until recently, the serology of the coli group was still quite confused. And here we are in 2026, and it is still quite confused. And the reason for this is that when you look at what we would have as a sort of typical gram-negative cell envelope, you have an inner membrane, an outer membrane. In textbooks, you often see it drawn like this. And you have, in the outer membrane, this lipopolysaccharide. This inside lipid here is the ligand for TLR4 that you heard about in the last talk. But this is attached to a very polymorphic polysaccharide called the O antigen. And this structure is highly immunogenic, so it's easy to make serotyping reagents against this thing. And this has been the basis of O antigen serology since the 1940s. This works beautifully, right? There's nothing confusing about this. You have an antibody against your O antigen, you can tell what O antigen is present. The problem is that, again, these are our happy, friendly E. coli. I'm using dogs as analogies here because the O antigen here, when you see it drawn in these types of diagrams, looks rather sort of sparse and well-behaved. In reality, it's more like the hair on this dog, right? So these glycans cover the surface of basically all bacteria, but definitely the Enterobacteriaceae. And if we go now out to our wild bacteria, most of them have these group 1, 2, or 3 capsular polysaccharide structures. And these are longer than the O antigen, so they coat also the O antigen. And they mean that your nice serotyping agents don't bind, right? So you can have the O antigen structure present, but the serology doesn't work. And the reason for this also is that these capsular polysaccharides are very poorly immunogenic in the context of whole cells. So you don't naturally make antibodies against them. The way you make rabbit antisera is you inject whole bacteria. You do not get good antibodies against capsules, so that the serology typing for capsules is terrible. And to a large extent, we simply stopped working on them because it was a pain to work on them, so we just sort of ignored it. And actually, most of the lab strains have lost capsules, so you don't have to worry about it. They're very expensive to produce, right? That's a lot of sugar that you're putting out onto your surface. If you don't need it, you just lose it. But they are the outermost surface of most of these wild E. coli, and they are what we need to target. So we realized it was actually going to be very important if we were going to make useful functional reagents to target these E. coli that we understood these capsules and we understood what they were actually doing there. So also just to kind of nail home, everything you see about E. coli that's published has an O antigen and an H antigen. The H antigen is the flagella. You will not see any Ks in these. So the K antigen is the capsular polysaccharide. We're simply ignoring it in most studies. So the good thing about at least the group 2 and 3 capsular polysaccharides is that they have a conserved part and a non-conserved part. So the lipid anchor, and this is an oligo-beta-KDO, it's a short sugar that links the rest of the capsule to the outer membrane. This is conserved in all group 2 and 3 capsules, and the enzymes that produce it are also conserved. So that allows us to pull the capsular biosynthesis gene clusters out of whole genome sequences, and they have various different arrangements of these conserved genes, but they're typically flanking the capsule-specific genes that actually synthesize the capsule-specific polysaccharide. And so what we did was we went and sequenced the capsule type strain collection. We called all of the K-specific genes. We then went into RefSeq with about 40,000 deposited E. coli genomes with plenty of metadata, and called also and then all of the novel K-specific genes. And the team who did this, who required huge thanks for the enormous amount of work they put into it, was led by Tim Keyes, who's a senior scientist in my group, a glycobiologist, now running a spin-out I'm going to talk about in a minute, together with a group of Shinya Sunagawa, who are fantastic bioinformaticians, and 2 very talented postdocs just going to start their own groups, Eli Kachake and Sam Miravet-Virdi. So what they did was they exhaustively went through all of these capsule gene clusters, assigned functions to all of the capsule-specific genes using either homology or structural homology. And this allowed us not only to identify the known capsule types, which you can see with structures here, but also to associate the unknown capsule types as having very likely similar carbohydrate compositions to some of the known structures, right? Because we have very similar gene content in these unknown structures. They're not identical, they're going to have different linkages and different modifications, but they're likely to be in the same family as these capsules here that are containing ribose and KDO. And what they could do out of these structures, so there's now 130 of these defined, was we could make HMM-based models, we could convert this into a bioinformatic tool, and you can now take any E. coli genome, or indeed a metagenome-associated genome, feed it into this tool, and it will give you some very nice predictive data with relevant statistics about how confident it is of what capsule your E. coli strain is producing. So this is now a really useful tool for doing epidemiology and doing studies of what capsules are out there and what we need to target. And just to give you a rough overview of how much unknown stuff we found. Interestingly, this varies over different sources. If we look in human-derived E. coli's from databanks, actually a decent fraction of the human isolates have known capsule types. So we've mapped a decent amount of the capsule diversity that you find in humans, but there's still around 20% of the strains that have novel, unknown capsule types. If you go down to wild animals or even livestock, we're getting up to 40–50% of the strains having novel capsule types that have never been described before. So in the best described organism on the planet, we have a lot of unknown things coming up that we don't actually know what they are. But we're now starting to map those, and that gives us a much better idea of what's present and what needs targeting. Drilling down into this, here we have metagenome-associated genomes, samples, whole genome sequences from healthy individuals, either stool or other sites, and then a whole variety of E. coli isolates from disease. And one thing that you'll notice here is that actually the urinary tract infection, bloodstream infection capsule types that we're pulling out are very, very similar to what you're finding in healthy individuals. And that fits pretty well with what we know, that actually most of these infections are spreading within an individual. You're already carrying the pathogen in your gut, and it spreads from your gut to another site and makes you sick. But as long as it's in your intestine, it's actually fine. And that's going to be interesting for later, that we can actually start to target these things in the intestine before they cause disease. But actually, in parallel to developing all of these bioinformatic tools, we also developed a nice way to make glycoconjugate vaccines from these capsules, which now renders them immunogenic, allows you to make good antibody responses against these capsules. Tim Keyes, the person who was running the bioinformatics study, has founded a spin-out called Bacsiva. I don't have a financial conflict of interest in this, but I would declare that this is a spin-out from my group. They're funded by Carbex to get this through to phase 1 trials, and they can induce very nice antibody responses now against the capsule structures that should protect from at least the invasive pyelonephritis and urosepsis part of urinary tract infections. But vaccines are not always the answer, and the mint green bit of this graph and the salmony pink bit of this graph correspond to Q2 capsules, the K5 capsule and the K1 capsule, that are actually identical to human glycans. So they're mimics of host. There are 2 structures here that account for a lot of infections that we cannot vaccinate against. And I particularly want to highlight the meningitis down here. These are all neonatal meningitis cases. The vast majority of these are caused by E. coli producing the K1 capsule. It's an alpha-2,8 polysialic acid. It's found in the brain. It's also found on a type of immune cell. So we cannot vaccinate against these, and we would not want to vaccinate against these. You'd get autoimmunity. So this then comes back to this concept that actually these bugs, although they cause invasive infections, they're also being carried around in the intestine of healthy individuals. And perhaps instead of trying to target these by intramuscular vaccination, we could target these by trying to get rid of them from the gut lumen. And we know that we can do this. So we've had several studies published where we use the selective pressure driven by high affinity IgA, again, something Barclay referred to clump bacteria together. It gives them a massive selective disadvantage in the intestine because they get flushed through much more quickly in the flow of intestinal content. And if we then add in a competitor strain into the gut, we can drive elimination of an E. coli within a couple of weeks. So this is done in a treatment mode. We can really clear E. coli from the gut. And this is all work done actually with Mederic Diard. This is data from Mederic's lab. But again, this was done with one of our friendly non-encapsulated E. coli's. To take this to our K1 E. coli's, we need to do something else because our antibodies are not going to bind as long as the K1 capsule is there. So what Mederic's group came up with is the concept to use bacteriophage steering. And phage therapy is something that people have been using for a long time. But the problem with phage therapy is you get resistance. And so what typically happens is you treat your bacteria with a phage here. If there's no phage, they grow happily. If you treat them with a phage, you kill most of them, but a few resistant mutants grow out, and those grow up to the same level as your initial wild type. But if we look at what's happened to those bacteria, actually the most common resistance mechanism to a phage is to lose the receptor for the phage from the surface. And if the receptor that the phage is using is the K1 capsule, then what you lose is the K1 capsule. So all of these resistant E. coli here have lost production of the K1 capsule. And what we were able to show is that you can combine treatment with these bacteriophage with our oral vaccination and niche competition concept to actually completely clear out E. coli K1 from the intestine of adult mice. So you can now really get rid of carriage of this bacterium from adults. And of course, this is then really interesting for can we prevent this from infecting neonates. And so we did the experiment also where we treated pregnant dams with the vaccination and with the bacteriophage and with the niche competitor here. We can suppress colonization of the pregnant mothers, and we also get pretty good prevention of transmission of this bacteria to the pups by day 10 of life of the mice, which is their early weaning period. Okay, so what I hope you remember that we've made significant progress from microbiome engineering for child health during this project. We have very diverse interactions with the microbiome, and really understanding what we want to engineer in different situations and different diseases is definitely as much of a challenge as the engineering itself. We think nitrogen handling by the gut microbiome is something that has been underexplored. We have now novel techniques to explore this, and we're waiting now for people to want to run the human studies and apply these techniques also to looking at microbiome handling of nitrogen in humans. Carriage of nasty antimicrobial resistant opportunic pathogens is very common in the intestine. It's a major source of infection, and we have now a process that should allow us to remove this source of infection. To do this, again, we really need to understand what we have to target on the E. coli. Just targeting E. coli generally isn't sufficient. We needed to actually understand these capsules. And being able to bring bacteriophage into the intestine, because it's topologically outside of your bodies, right? It's not in your tissues. It doesn't matter that there might be an autoantigen that might bind a bacteriophage. The bacteriophage are not going to do anything to your own cells. We can actually have a safe treatment that allows us to get rid of K1 E. coli from the gut. And what we're aiming to do now is really to translate this into clinics. Of course, one of the big challenges in paediatric health is that we're not going to run a first-in-human trial in neonates. So we're initially going to look at decolonizing pandemic, multi-drug-resistant ESBL E. coli strain ST131 in adults, with the aim to then eventually moving this into maternal treatment to prevent transmission to neonates. So with that, thank you for listening, and I look forward to your questions.